So welcome to today's lecture, which is Your Child's Brain on College in an Unusual Time. And we're going to talk about how nature and nurture collaborate to develop thinkers. My name is Maria Donahue-Valleka, and I am the brand new Dean of Arts and Sciences at William & Mary. I am not teaching yet, but I will soon. And so this really is an opportunity for me to teach parents first, and then convince your kids to take one of my classes in the future, because I will be teaching every semester from here on out. I am a neuroscientist. I have spent my scholarly career figuring out the molecules that help cells of the brain become different from one another, and then how those differences affect function. So let me map out for you what the goals of today's lecture are. First is a little introduction to the nervous system. Second is to tell you about how the brain can change over time. And finally, to give you comfort that your son and daughter's brains are still changing, and just a reminder that so are all of yours and mine. So we are currently in flux. The job of the nervous system is to drink information in from the world through our senses, and that is through smell and taste, vision, hearing, and touch. And of course, each one of these pieces of input from our world come in lots of different channels. We call that input, the sensory information, afferent information that goes into the nervous system, the spinal cord and brain. In the brain, there is a lot of integration and processing, and there is then an output created. We call that efferent information, and that is the response to the environment. It could be a cognitive response, it could be motion. And a reminder to all of you that the basic unit of the nervous system is a special cell that is called a neuron. It is a polarized cell, so that it is different at one end than it is at the other. And information in a neuron flows in a single direction. It flows from the head, or what we call the dendrites, which are these sort of spiky, hair-like extensions from the cell body, down a very long axon to a nerve terminal. And in this case, this is a chemical neuron, so it's spitting chemicals that are here in the nerve terminal onto the environment, which is the next cell downstream. And when we think about neurons, we think about them in circuits. And so that is that your nervous system only works because one neuron, now drawn much less beautifully than the last slide, is connected in a circuit to a second neuron. And the point at which these two cells communicate with one another is called a synapse. So in this circuit, we call this first cell the presynaptic cell. It is the cell prior to the synapse. And this second neuron, the postsynaptic cell, it is after that synapse. But of course, in our nervous systems, there are many cells. And so let's imagine that those two cells have a synaptic partner down here. There's another synapse that exists between the second cell and the third cell. And now in that case, the definition has changed. This cell is now the presynaptic cell in relation to that synapse, and this is the postsynaptic cell. And so we have circuits that operate and underlie every single function, and they are loosely operated like this. Okay, so here is a little bit of an animation where this individual is hearing information, it's being integrated by these brown neurons, and the output is through the blue. So let's watch that again. Input is coming through auditory information, activating a first cell, then activating second brown cells, and eventually leading to output of a motor neuron, something that would, for example, move muscle. So that seems pretty simple. What's actually happening, okay, let me get this going. If I could have a little help advancing, there you go, thank you. So what I drew for you previously was a very simple circuit. This is actually what circuitry looks like within your brain. This is the visual system. So down here are the retina, which are your detectors, your photodetectors of vision. Those retinal neurons then send information into the thalamus, into the cortex, and then in this wiring document or diagram that we have here, every line represents information flowing to a location in the brain, but also back from the location in the brain. So you can see that this circuitry is really incredible until it finally arrives at a part of the cerebral cortex where we might produce motion from that visual input that happens down here. About 40% of primate brains are interested in vision, are delegated to vision in humans. Really interestingly, of course, mice don't have great visual systems, so their brain is largely devoted to somatosensory, the sense of touch of their bodies and whiskers. So this is what we are dealing with when we try to understand the complexity of the brain. What we're trying to do is break apart circuits that look like this. So a big challenge. And we do all of that because we want to understand human behavior. So I sort of pulled this from a review of a buddy of mine. So there's lots in here that we don't need to talk about. But why do we study the brain? We study it because we want to understand how people make decisions. What are mistakes and learning? What is, self-control? For some of us, what is procrastination? There is so much that is fascinating about the brain. For example, here, the placebo effect is a real thing, and there is a neural basis for it, and it is fascinating. And again, my pitch is send your kid to my class, and we'll talk about it, and then over Thanksgiving dinner, when he's home with you or she, you can have that conversation with them. So there's a lot of information in the last 15 years about the role of a genome, of what our DNA does to impact our behavior. And in order to impact our behavior, of course, now you understand we need to impact brain structure and function. So the question is, are we a reflection of our genes? Let me start with reminding you what a gene is. First, what a genome is. So this is a chromosome smash. This is just pulling a little blood out of an individual, getting rid of the red cells that don't have nuclei, and then smashing open the nuclei of those cells that remain. We have 23 pairs of chromosomes in human cells. Unfortunately, they're not color-coded like this. These are, scientists went in and did pseudo-coloring on that. We can take any one of those chromosomes, and now very quickly we can sequence that. And so here is a sequence plot. I hope it's coming soon. Yep. So this is a little stretch of DNA, and each one of these colored peaks represents a single nucleotide that makes up that sequence. And so we can take any chromosome, and we can figure out exactly what the component sequence is. And then we can name genes. So here is a chromosome sitting here. Here's the centromere. Here's the short arm. And here is the long arm of this chromosome. And each one of these different lines that are hard for me to read, and maybe for you too, represents the notation that there is a gene that exists in that location. And we can identify those genes. We know what they do pretty much in most cells, although there still is so much that's unknown. So more than 20% of our genome, we know there's a gene there. We just don't know what it does yet. Okay, so if we look at a chromosome, so again, there's the centromere, there's the short arm, and here is the long arm, and we spread it out, and we look just at a single string of DNA, what we know is that there are regions that we can identify as genes. So that is a stretch of DNA that makes a protein, and that protein does something inside of cells. And then there's a whole bunch of intergenic regions, or just what we always thought of when I was training as junk. But we know that they're not junk anymore. We know that they are actually really important in regulating gene function. So for the purpose of this, we're looking at gene 1 here and gene 2 here, and then asking how they act, and then what they do to affect nervous system function. And so I don't have to remind any of you of the power of genetics. Here are identical twins that, at least by their account, should have made billions of dollars on Facebook and yet didn't. But these brothers are identical twins, and you can see that genetic runs pretty deep. So how is variability determined if we are genetically dictated individuals? When I was in high school, and maybe for some of you parents too, we learned about evolution in two different ways. there was a guy, an evolutionary biologist named Lamarck, and Lamarck believed that environment affected the features of organisms. So here are giraffes, and let's say this giraffe started pretty short, and because he couldn't reach the food, he adapted to that environment and caused his neck to stretch, and so he got taller in his own lifetime. And then, again, pass that on to his progeny. And this, when I was in high school and college, was scoffed at. It was a crazy idea that environment had any role in the features of an organism. And that's because Darwin came around. And so what Darwin showed, and these are the finches that Darwin studied in Galapagos and many other places, demonstrated that there is great variability in the features, or what geneticists call phenotypes, of different organisms, and that the survival of the fittest is that the features that are most adaptive in a particular environment will be selected for, and those that are non-adaptive will be selected against. And this kind of variety that he saw in finches, and we now know to be true across many, many organisms, was really natural selection, that it wasn't adaptation in the moment, it was selecting for traits and then passing those traits on. And so again, when I was training, and probably many of the parents here, Lamarck was crazy and Darwin was the story. It's not so easy as that. Usually in science, the fun of being a scientist is that you think you know what you know, and then shortly after, you don't. So what we learned over time is that genetics are not the whole story. So for example, these are two mice that are siblings to one another. They are genetically identical to one another. These mice are called agouti mice, or OB mice. And this mouse line was identified at this fantastic mouse facility up in Maine, Jackson Labs, where this line of mice arose spontaneously. So these mice are genetically identical to one another, but you can see that the fur color is different. And the fur color is actually a great marker of obesity. So these are mice that become obese over time. And the question that scientists had was, why are these mice getting obese when their genetically identical litter mate is not getting obese? And this led to a whole new field called epigenetics that people were unprepared for, but has really informed the way that we think about different features of organisms. And what I will get to shortly is it also informs us about learning. Okay. So here is what we know happened in those agouti mice. Let's say that there is a gene sitting on a chromosome. This gene is unmethylated, so it's not modified by a chemical moiety that is here, this sort of little stick figure. So this is an unmodified gene. When this gene is unmodified, it can be expressed, and so the gene is active. And when the gene is active, there's a lot of gene expression. In another situation, that very same gene will have these modifications, these methyl groups that sit on the DNA. And what that does is it eliminates the ability of that gene to be active. So that gene is inactive. And when that gene is inactive, there is less gene expression coming off of that gene locus. And what we've learned about this is that environment can cause the addition of methyl groups, or actually the removal of methyl groups, to dynamically regulate the ability of a gene to be active or inactive. So here is a female mouse of that agouti strain that I talked about. And this is an unmethylated mother, and so she has active gene expression. If that mother, while she were pregnant, was giving no dietary supplement, then 3/4 of the offspring of that mother would be yellow and would eventually become obese. But if, on that same mother, we supplement her diet with extra methyl groups, so we actually provide in her food something that will allow the methylation of genes, then what we'll see is that 3/4 of the offspring in that case are brown and are of normal weight, and that is because this agouti gene is methylated, and when it's methylated, it's silenced. So this was one of the first examples that this ability to dynamically change in real time in a single individual, not transgenerationally, we could change features of individuals. And we know that it's not just that situation of agouti that happens. We know that when an individual is stressed, the genome of that individual will become methylated. Again, when people are anxious, the genes could become even more methylated. And what is fabulous and unexpected is that this can be transgenerational. So this woman is pregnant. She is methylating the genes within her offspring. That offspring here is then pregnant, methylating the genes, and this granddaughter of the original will have changes in their gene expression. And what was really striking, there was a fantastic study a few years ago where there was a group of individuals who lived in a very mountainous area that was very isolated, and so it was a great place to study genetics. There were repercussions in the grandchildren of whether the grandparent happened to live during a time of feast or famine. And so the metabolic activities of a grandchild were actually most well predicted in this case by what the lived experience of their grandparent was. And this is called transgenerational epigenetics. And so that is that it's not just methylating genes in an individual's lifetime, it's methylating genes all the way through lineage. Okay, so academics like me read books because it's fun to find new ideas. And so I am a member of a group of intellectuals who every year come up with what is your dangerous idea. And this is a book that was probably from around 2005, but still it's an idea that stuck with me and one that I'd like to share with parents today. And that is that there was a psychologist who, quite frankly, is not the most well-regarded psychologist in the world. Yep. Okay, right there. And she wrote a book that is called The Nurture Assumption. And her dangerous idea that was presented in this scholarly sort of post is this. Is it dangerous to claim that parents have no power at all, other than genetic, to shape their children's personality, intelligence, or the way he or she behaves outside the family home? And so I think for any of us who are parents, we think that we bring value to the lives of our kids, and that we can impact the way they see the world, what they value, and the way they behave. But I think we also know, I have two sons, and each one of them is their own person, and they were their own person from the very earliest times that I remember who they are. So this idea to push against is how much can other people really impact one another's lives. And what I tell you is I think that they impact one another's lives greatly. So let's get back to that genome. And what I just want everybody to remember is that every cell in your body contains the same DNA. But each cell uses a subset of the whole. And this is so important to me that I'm going to take you through sort of the mind logic here. Okay. So again, here is a cell. The yellow circle is the nucleus. The little X's inside are chromosomes. There's the chromosome. You can unwind it, and that's DNA. That DNA, when expressed, will make proteins, and those proteins do stuff in cells either on their own or they aggregate into groups. So understanding what proteins are expressed in a cell are super important for understanding what that cell is capable of. So your genome is about 25,000 cells. Your genome is 25,000 genes. And of those 25,000 genes, we can class them into different groups. So for example, there are always a bunch of genes that we call housekeeping genes, and these are genes that are present in every single cell that is alive. these genes are expressed to make more DNA, to make RNA, to make proteins, to make more membrane, to correct errors. These are the genes that are present uniformly and ubiquitously across mammalian cells. And then let's say that you could take some portion of that genome of 25,000 genes and say, okay, these are the liver genes. These are the genes that make proteins that detoxify the blood. And so when your kid goes out and has super fun time on a Saturday night, doesn't feel great the next day, that is because they blew out parts of their liver, and they are now recovering those function by activating these genes and repairing any damage. Similarly, there are lung genes that are expressed selectively in lung, and these would be the genes that are responsible for the transport of gases across the lung and removal of carbon dioxide and bringing in oxygen and all of the energy that is related to that. And then there are a bunch of genes that are expressed in the brain. And because the brain is so important and so complicated, more genes are expressed proportionally in the brain than any other organ in the body. So if we take those genes that we know are expressed only in the brain and we divide them up, we know that the neurons that I talked to you about are right here. In the green circle down here are other cells of the brain called glia that serve support and actually active signaling, partnership signaling with the neurons. And then what's shown here are these ovals of other kinds of, within the neurons, of subsets. And so that is because neurons are not one thing. So these are pictures that the famous neuroanatomist Ramón y Cajal drew over 100 years ago. And just for the record, you can't actually be a neuroscientist if you don't show one Ramón y Cajal slide in every lecture that you give. So these were basically tracings that he saw under a microscope of different cells, and you can see that even though these are all neurons, they are remarkably different. This is super skinny and simple. These are very complicated. And so the fun for neuroscientists is that once you find a cell, you can name it. So this is a cartoon of some of those cells. So this red guy right here, you can see is shaped like a pyramid, and we call that a pyramidal cell. It's one of the most important cells of your cortex. Pyramidal cells are the reason that you can move around with voluntary motion. Here's a chandelier cell, a basket cell, my favorite, the double bouquet bitufted cell, a Martinelli cell. So you can see that there is a lot of variety in the shape of cells in the brain. And then just trust me when I say that that variety, the form of that cell, reflects the function of the cell, and the function of the cell is going to drive very specific neural responses. And so just to back up what I said about genomes, so what you should know about those cells that I just showed you, and I'm having a little trouble flipping back and forth, not sure exactly what happened there, is that each one of those cells that are shaped differently will also express a different subset of genes, and that is what makes them special. So what's learning? Let me move back to among the simplest systems in the nervous system, and that is the neuromuscular system. So here, drawn horizontally, are muscle fibers that are, of course, much longer than this. And here there are 3 motor neurons, and these would be sitting in the spinal cord, that really early in development, so perinatal, right after birth, will come into the muscle and will start sending out shoots and sort of, forming a relationship with what they will eventually form a synapse. And neuromuscular junctions look like this, these little blobby nubs at the end of the axon. So perinatally, there are 3 motor neurons here, and they are all sort of talking with different muscle fibers. There's not a lot of coherence here. And so this kind of disorganized connections between these 3 cells and the target is why infants and toddlers are pretty clumsy, because they don't have coherent synaptic connections yet between the motor neurons and the muscle fibers. Over time, what will happen in this system is that some of these connections will become strong, and some will become weak, and when they become weak, they get eliminated. So what ends up happening in adulthood in many of you, and especially in super coordinated children that we have, is that you can see that this blue cell comes in, the axon branches out and forms a connection with 3 muscle fibers. That's the only connection that that muscle fiber gets, is blue input. The green neuron comes in and branches off and forms a connection here and a synapse over here. And so again, these 2 muscle fibers are only receiving one kind of input, similar for the yellow. So what's happening over time in this very simple system of neuromuscular is that there is a refinement of the synapses. And what we're basically doing is we're eliminating the number of synapses that happen over time. And so again, from that really clumsy toddler to the super coordinated adult, what we're doing is we're actually limiting the number of synaptic connections. But in doing that, we're making those connections much more effective and leading to great function. Okay. Okay, so what's learning? Learning is taking that incoherent innervation, and this is right after birth in a mouse, and this is now a part of the central nervous system, and over time, eventually rearranging the way that the neurons talk to one another, so that by the time, this is now postnatal day 15, and this is in mice, there is a reorganization of the cell-cell contacts that then leads to great function, quick, accurate, reliable function. And this progression of a sort of incoherent connection between different neurons to a very precise and specific connection is what happens with learning. Okay, so what does studying for your student, or for your children and my students, do to the nervous system? Really what it does is it builds a pattern of connections that work. So once a neuronal connection, a synapse has been used, and it's been used well, it will be more available to that individual the next time. So for me as an instructor, I give homework. I ask my students to read. I ask them to puzzle through hard questions. And that's good because there's some content there and they should learn that. But what we're really trying to do, what I'm really trying to do, is get those neural circuits working so that they can be activated in specific ways. And what we know is that when a student has a hard problem once and they activate a circuit, when they have another hard problem in the future, that circuit will be more available to them and they will be able to solve a problem more clearly. So this is a picture of a mouse brain, and these are different kinds of neurons, and what it's showing here is that what we're doing in our life is just exercising circuitry. And that's what learning is all about. Okay, so one of my favorite quotes as an educator, and I have to say as a parent, is that good judgment comes from experience, but experience comes from bad judgment. And so part of learning is also, it's knowing what you want to do, but it's also learning what you don't want to do. And that is reflected in the neural circuitry, that when there's a bad outcome, that synapse would be eliminated or made weaker. Okay, so I just want to point out that there's so much cool stuff going on in neuroscience. This is an image that actually the person that I got my PhD with created. It's called Brainbow, and it's a way of introducing individual colors to specific neurons so that you can see what diversity looks like. And then there's a great NIH-funded program, which is the Brain Research Through Advancing Innovative Technology Initiative, which we're learning so much about the brain from. So what we're really looking for as neuroscientists is what's our connectome? What are the pathways that we use to create particular functions, and how can we strengthen those so that they are most effective? And something that's really important to remember is all of you read and I read, but the way we do it might be just a little bit different. So we all perform similar functions, but we may be unique in the wiring that underlies those functions. And just a reminder, and this is, I think, my second to last slide, that over time, what will happen is when one cell talks to another cell and that communication works out well, it will strengthen that, either by branching off and forming more connections, or in the case here, sort of there's a battling out of two different synapses onto this cell, and the most successful connections will then become strengthened and the others will be weakened. And that's happening throughout our entire lives. Even in old people like us, we are still changing our synaptic connections and able to obtain new functions, no matter what your kids tell you. Okay, so the goal of the nervous system is that there are neuronal best friends forever, but it's only for a while, and that we are constantly rejiggering and modifying our synaptic connections. And then this is just a reminder that because I am a molecular neuroscientist, all of that is related to molecules that are expressed in these cells, and those molecules then will guide function. And again, I'm not going to talk about it here, but send your kid to me when I'm teaching a class. Okay, so do personal interactions matter? And what I would say is, does this moment that we are living in matter? We are under pandemic. Our kids are being asked to do things that they've never had to do before. So what do we know about that? We know that in cases of people, twin studies, identical genetics, that there are still differences. We know a lot about abuse and neglect and the neural changes that will happen. We know about maternal deprivation. And then really happily for neuroscientists, because it's not always great to study the stuff that's not working, we also know about connectedness studies. And so for people who are especially connected, what that does to your brain, mindfulness, whether it's meditation or praying or things like yoga, definitely releases chemicals in the brain and changes neuronal circuits. Stress is actually a physiological response to the environment. It's part of that drinking in from the environment. And we know that stress can dramatically change neural function. Inequality and justice. There are some really awesome studies that demonstrate that cognitive ability is impaired by poverty, for example, and that it's not the other way around. And then finally, vulnerability. And I think it's something we all have to remember in this moment of pandemic, is that each one of us has our own sense of risk and safety, and that also will change neural function. Okay, so what I want to tell you is, as far as I'm concerned, all of you are the heroes of today. You have accompanied your children for a long time, and we are super glad that you gave us the privilege of being part of their lives too. So this journey that you have walked them on up to this point, we are so happy that we are partners with them now. Just remember that even though you're my hero, you might not always be their hero. And so this is, I'd like your advice. Why are you asking me to tell you how to live your life? And then it's because I want someone to blame. And so I think that is an age-old relay and is not going to change under these current conditions. But what I'm always reminded of is the Michael Jordan quote, which is, I failed over and over, and that's why he succeeded. And so part of being a teacher for your kids, part of you being a parent, is watching them fail, watching them do something and it wasn't great, and then they learn something. So it's that bad judgment helping us form good judgments. And so finally, I just want to point out this is the Center of Arts and Sciences at William & Mary. It's called Ewell Hall. It's likely to be called something else soon. I am so glad to be spending this time with all of you and really hope that I get to meet you all soon. Here is my email in case anybody wants to follow up. But again, thanks for being part of Parents Weekend. Thanks.